Hello, everyone. My name is Mark, and welcome to this new episode of the mitoPodCast. For those of you joining us for the first time, the mitoPodCast is a new program that we hope will build awareness in the mitochondrial medicine, research, and patient advocacy community on topics related to mitochondrial disease and health, as well as to highlight cutting-edge research in the field. Our audio format episodes are approximately 10 minutes in length and will feature the exciting work of an expert in the field in a way that can be understood by all in the community, regardless of educational background. Included with every episode will be an infographic summary of what was discussed, so please check that out as well. Today, I am delighted to be joined by Dr. Mark Tarnopolsky. Dr. Tarnopolsky is a professor in the Division of Neurology at McMaster University's Departments of Pediatrics and Medicine. He is also the Clinical and Research Director at McMaster University's Corkins Lammert Family Neuromuscular and Neurometabolic Clinic. Dr. Tarnopolsky has authored or co-authored more than 400 scientific articles and has lectured frequently in the area of neuromuscular and neurometabolic disorders, aging, and physiology. He has served on several editorial and scientific boards, including for the United Mitochondrial Disease Foundation and the journal Medicine and Science in Sports and Exercise. He has been on numerous grant selection committees for the Natural Sciences and Engineering Research Council and the Canadian Institutes of Health Research. Dr. Tarnopolsky, it is my pleasure to welcome you to the mitoPodcast.
Thanks very much.
Great. Thank you for taking the time out of your busy schedule to meet with me. So, Dr. Tarnopolsky, your research program is well known for its efforts to find nutritional, exercise, pharmacological, and genetic therapies for primary mitochondrial, neuromuscular, and neurogenetic disorders. Could you explain more about the background of your research program to the listeners?
Sure. I've been in the area practicing as a clinician for just over 25 years. And although we've all hoped that there was going to be genetic therapy for many of these disorders, it was clear that we had a long way to go, and therefore we had to come up with something to help our patients. I've been very interested in the multisystemic benefits of exercise and how nutrition can influence that as well. And it was pretty clear that if we could get people to exercise, we might be able to improve things. And certainly over the years, we've shown, especially with muscular dystrophy, some of the benefits, mitochondrial disease as well. Pioneering work from Tanya Tavasalo and others have clearly shown the benefits of both endurance and resistance exercise. So we've been heavily involved in that aspect of things, and to also look at how nutrition can enhance some of the benefits. And we've studied this in young, healthy individuals, older adults who have both muscle weakness and mitochondrial dysfunction. And then we've applied it in animal models and eventually to humans. That's resulted in one of our compounds, if you will, where we looked at 4 different nutritional compounds: coenzyme Q10, lipoic acid, vitamin E, and creatine, which we found was beneficial in our patients with mitochondrial disease to lower some of the biomarkers. So those were ancillary things that we thought that we could do to improve people's quality of life and their daily functioning. Ultimately, the goal, however, would be to come up with a cure. Certainly from a nuclear genetic perspective, we're slowly starting to get closer to that with some of the viral gene-based therapy deliveries. And I think that's certainly one area that I think in the future might really have a huge impact. We started down that pathway looking at the potential for exosomes as a therapeutic strategy. And we were hoping that some of the benefits of exercise might be mediated through exosomes. And indeed, they may. But we do have a paper coming out where we found, first in a MELAS patient, when we did a bone marrow transplant, we actually didn't see any of the beneficial new mitochondria get transplanted into the person's tissue. So we kind of shied away from that, and we're still focusing more on some of the nutritional stuff. I think there's a lot that we still need to do in this area, but for now we're predominantly still trying to expand on our exercise benefits.
So, Dr. Tarnopolsky, could you explain to our listeners some of the findings of your research into mitochondrial myopathies, and how do you see this translating into the development of novel therapeutics?
Right. Well, we could take the example of some of the things that we've tried, and I'll explain how that's relevant, I think, to development of future therapeutics. back in 1999, I think it was, we found that creatine levels were very low in the muscle of patients with mitochondrial myopathies. So we did a clinical study shortly thereafter showing the benefits of creatine improving high-intensity exercise performance. We also know that creatine has a neuroprotective effect, so I think in the long run it's probably helpful from a stroke perspective in patients with MELAS syndrome and other mitochondrial diseases that predispose to strokes or other energy deficiencies in the brain. However, the concept of multiple final common pathways, was very apparent to me back then, and sort of formulated our move forward. So the next thing that we tried to do was to target some of these pathways. So we came up with a combination strategy with the CoQ10, the lipoic acid, the creatine, and the vitamin E to target many of the final common pathways, and showed that was helpful, especially in the MELAS patients, to lower the markers of disease. So lactate and oxidative stress went down. And I think as our group and others move forward, I think we really need to be thinking about combination strategies. And I don't think that there's going to be a single molecule or therapy, other than perhaps a gene therapy, if that proves to be successful, that's really going to work. I think we have to combine our resources and target some of the multiple final common pathways, very much like cancer therapeutics, where if you use a single compound, a chemotherapeutic agent, you can get some improvements. But when you look at how we now treat acute lymphoblastic leukemia with multiple drugs coming together to target the multiple final common pathways, and the success in that. So I think that's what we really need to do as we move forward, and something that we're trying to do ourselves is to use combinatory strategies to develop some therapeutic interventions.
Great, thank you for that, Dr. Tarnopolsky. So I think you touched on this in the last question, but could you explain to listeners some of more about maybe some of the next steps you'd like to take your research program?
Yeah, I mean, one of the things we've been looking at was expanding even more on this concept of using a multi-nutrient approach, and that's one of the things that we're doing. So we started with our 4-nutrient composition, and we wanted to see if we could further improve mitochondrial function by adding other things to it. So people have found that beetroot extract, which contains nitrates, actually improves mitochondrial efficiency. Some other compounds increase the total amount of mitochondria, but we thought it might make sense to take our mitochondrial 4-component compound that we came up with and add beetroot extract to see if we could further improve mitochondrial function. So we've done some studies in the obese mouse model, which has secondary mitochondrial dysfunction, and showed that yes, when we added the nitrates, it further improved mitochondrial efficiency. But we also had 5 other components that we were using more as a weight loss strategy. But when we mixed the two together, we found that all 10 of them together, somewhat surprisingly, had a very significant beneficial effect on the mitochondria and white adipose tissue. So that was interesting because we really upregulated mitochondria in the white adipose tissue and pretty much reversed the obesity in the mice. So then what we've done is tried to subtract out some of the components to see what the essential core of these were, and have come up with a 7-constituent or 7-component supplement which activates a Browning program in white adipose tissue. It has small effects in muscle, but a very potent effect in the adipose tissue. So certainly for Murph syndrome, as an example, where they get massive lipomas, so the white adipose tissue grows and you get these huge lipomas in the head and neck, that could be a very targeted, specific therapy for a disease like that, where the white adipose tissue is affected by the mitochondrial disease. And I think combining a strategy like that, where it focuses on one tissue, and then other strategies which might work more on muscle or brain tissue, and bringing them all together might be an effective therapeutic intervention, which is something we're working on.
Great, thank you for that, Dr. Tarnopolsky. So the development of new treatments for disorders involving mitochondrial dysfunction is not easy. In your view, how can we accelerate the therapeutic development process in a careful and responsible way that prioritizes patient safety? Also, is it even possible to achieve this balance of ensuring patient safety while developing drugs quickly?
Yeah, there's several components to that. I think one of the ways to accelerate this is to work together. We all have different areas of expertise. And when you work together and we have frequent meetings, sometimes, we might have an idea and we think that we're going to try this or try that. And someone else says, oh, I've already, done work, but I just didn't publish it. And that's not going to work. I think if we work together, we can certainly, keep some people from going down rabbit holes, which have already been explored, but maybe not published. And I think that's why working together and being open and collaborative is really the only way, I think, to achieve these things quickly. And it also saves a lot of resources, because someone might have a grant that looks fantastic. They get a whole bunch of money from Mito-Canada or UMDF or CIHR. And somebody in a room, if we're all working together, says, oh my goodness, that's useless. We've already done that. So I think that's one way that we can, more effectively focus people into one area, and also to work synergistically. Certainly sounds pompous to think we actually had a good idea, but some of the best ideas I've ever come up with is when I was working an area that was not my main focus. One of them, for example, I was doing my comprehensives in an area of inositol triphosphate and looking at lithium in the area of psychiatry, and saw that it influenced the inositol triphosphate pathway, and then saw the link between that and skeletal muscle fatigue, and actually published a paper and won an award based on a presentation that I did on that. And again, it was completely an area outside of where I was focusing on, and it was more from psychiatry and the use of lithium that got me thinking about it. So I think when we bring people together from different areas, that's going to really be beneficial. And as we move forward, I think it's critical to have good animal models, because certainly, we have to do preclinical work, and that's certainly from a rapid turnover perspective. We can screen a number of things fairly quickly, and that's always been a bit of a challenge in mitochondrial disease, especially with the mitochondrial DNA mutations. For complex reasons, it's hard to get mice models. But we are starting to get the POLG mouse, and there's some Lee disease mice, et cetera. And I think that's going to accelerate things quickly. I think now there's enough people also who have experience in clinical trials with mitochondrial disease that we can facilitate the development. So, for example, the STEALTH trial with their compound, there's a lot of time, a lot of expert opinion as to what might be effective outcomes. And I think now we have much better consensus on what the outcomes might be for clinical trials. And I think that's going to help us to bring things to market safely and effectively.
In your view, are there any novel therapies for primary mitochondrial myopathies that you believe shows significant promise right now?
Yeah, it's a difficult one at this point, because there's a number of strategies that people are looking at, trying to increase mitochondrial biogenesis, activate autophagy, and I think one of the things that we need to think about as we look for therapies is to have a gold standard. And certainly when you think about a gold standard for mitochondrial health, we think of exercise, and it activates autophagy. It ultimately downregulates oxidative stress by increasing antioxidant enzymes, increases mitochondrial biogenesis, increases mitochondrial turnover and fusion. So comparing things to exercise, I think, is one way that we can ensure that something's moving down the proper pathway. Certainly the mitochondrial transplant is of some interest. And there's definitely been success, especially from the cardiovascular perspective. Practically ramping up the mitochondria, delivering those mitochondria to an entire tissue is going to be a challenge. Sorry, I should say to an entire body. If you deliver it just to a small tissue like a myocardial infarction, I can see from a volume perspective how that might work. And there's been some evidence that might be of benefit. It's going to be a challenge, however, to get mitochondria into the brain, for example. And as we know, and certainly in our clinic, most of the biggest challenges we have are seizures, stroke-like episodes, ataxia, balance issues, and crossing the blood-brain barrier with anything other than small molecules or viral-mediated therapies going to be a bit of a challenge. But certainly there's a lot we're going to learn from the mitochondrial transplant work. And we have that paper coming out in the FASEB journal within the next month or so, showing certainly from a bone marrow transplant perspective, we can't deliver mitochondrial DNA in any meaningful way to the human body, which is a little disappointing. But, one step forward, one step back, but we've got to keep moving ahead.
And for this next question, I'd love to get your perspective on this. So from your perspective as a research scientist and a physician, how do you think a network of patients, researchers, and physicians collaborating together benefits mitochondrial medicine and research? So I guess in other words, how can patients benefit from those of diverse backgrounds coming together to solve pressing issues in our field?
Well, I think there's no way you can move forward without doing things in a collaborative fashion. If you do, you're going to be making a lot of mistakes, and it's going to take 3 or 4 times as long. There's no question that, and we've seen this with United Mitochondrial Disease Foundation, that having patient involvement helps, because sometimes we as physicians or researchers think that weakness or ataxia or whatever might be the thing that's most important to the patient, but we need them to inform us what are the issues that are most important to them. How's the disease impacting their daily life, and what do they see as a meaningful outcome? So certainly that helps us to choose outcomes that we think are meaningful to the patient population. The physician, it'd be very difficult without a strong knowledge of basic science to come up with things that are particularly novel. It does happen, but I think that both people have to work together. And the flip side is sometimes a basic scientist who's working in a test tube might not think about the physiology of delivery, for example, blood-brain barrier mechanism. It's easy to spike things in and use electroporation or whatever to get RNA into a cell in vitro. But practically, what are the barriers going through? And certainly the translational researcher, the clinician, might be a little more aware of those things. Working together, I think, is the only way that we can really have things move forward effectively. And also, not go down a rabbit hole, because it may be that in a petri dish something makes a lot of sense and a researcher gets millions of dollars to do research, but there's absolutely no way it can be practically translated to an effective therapy for a patient. Certainly, clinician scientist, a clinician might say, oh, well, this is just never going to work. Great idea, but practically it won't work. So I think that people really need to work together if we want to do this quickly and effectively.
Wow, thank you for that, Dr. Tarnopolsky, and thank you for coming on the program today. Do you have a final message? For our listeners, and how can listeners contact you if they have any questions about your work or would like to learn more?
Yeah, my email is publicly available. If you just Google my name and McMaster University, my email's there for the scientists and others to contact me, as they do every single day. As long as it's not too crazy and long, the questions, we're happy as quickly as we can to answer them. And I've had great collaborations just through email contacts and published papers with folks in this way, which has been fantastic. I think the final message to patients is, there's a lot of people working in this area, and there's a lot of interest in primary mitochondrial disease because people realize that mitochondrial dysfunction is something that every human being in the world has, because we're all aging, and aging is a mitochondrial disease. Obesity, type 2 diabetes all have components of mitochondrial dysfunction. So with that understanding, there's an increased interest in understanding mitochondrial biology, and then the spin-off from that is going to be therapies that we hope are effective for our patients with primary mitochondrial disease. So I've certainly seen a huge turnaround in the last 5 or 10 years where people are turning their attention to primary mitochondrial disease, even if the main focus will be to apply that to diabetes or obesity or these other things that are more common. The patient with mitochondrial disease is ultimately benefiting from the increase in all these great minds focusing in one area. So it's an exciting time. I certainly hope that for mitochondrial disease, we start to get breakthroughs like we see in spinal muscular atrophy, for example, with both gene-based therapy and antisense oligonucleotides, dramatically changing the outcome for especially the neonates and the young children that we're treating with these things. It's a life-altering therapy, and I really hope that that's what we start to see for mitochondrial disease.
Great. Thank you so much, Dr. Tarnopolsky, for coming on today and for discussing your innovative work. Before we end this episode, I'd like to thank all our listeners for listening to today's episode. If you have any comments or suggestions for future episodes, we would love to hear from you. Please head on over to our website to download our episodes and to see the upcoming events we have for this year. We have some exciting initiatives planned, so please stay tuned. Thank you all once again, and tune in to our next episode featuring another expert doing exciting work in the field of mitochondrial medicine.